Good evening once again, and welcome back to SBT 214, Biomembranes and Enzymology. We are continuing our course today with membrane lipids. So we are done with the enzymology part. We are moving on to the biomembrane part. And we'll start with some revision. I think that you would have done in some biochemistry in the first year already, looking at the structure of lipids. But we'll remind ourselves of some of those things that you studied in first year on the structure of lipids, because we are going to build on it when we come to the structure of biomembranes, before we continue to look at the various functions of biomembranes and various other aspects of biomembranes. So today it's more like a revision of the things which you have learned about lipids. Perhaps some new things will be added, and then some things which you didn't have the opportunity to have a better understanding of. You have another chance so that you can understand these things much better before we continue to look at more complex structures of biomembranes. So first of all, what are lipids? These are what you simply refer to as fats and oils. And the simplest of the lipids that you come across are the fatty acids. It's just like how we say that the building blocks of, say, carbohydrates are monosaccharides. Building blocks of proteins are amino acids. The same thing when you talk about lipids, the building blocks are the fatty acids. Now, what are these fatty acids? So fatty acids, when you look at the structure of fatty acids, you see there are two major parts of this structure. You have the carboxylic group. So, or sometimes you say it's a carboxylic acid group. Carboxyl group is understood. The COOH group, that's a carboxyl group. Of course, that is the group that gives it the name an acid. Fat, so the fat part is the hydrocarbon part. So you have this long hydrocarbon chain, which you come to see why is we say it's fatty. So right from the name, you can derive that, this is why we lipids will be, say, insoluble in water or anything like that. But we'll come to that and we'll see it much more. So these are the basic units of the lipids, fatty acids, which you can see the carboxylic acid group and then the hydrocarbon chain that is attached to that carboxylic acid group. Now, looking at that structure, the fatty acids are referred to as amphipathic structures. And what does it mean when we say a structure is amphipathic? So amphi means two. Just like you have, say, amphibians. Amphipathic it means that this structure has a part of it which is hydrophilic, is soluble in water. So that part of it can have interactions with water molecules. And you have a part of it that is the long hydrocarbon chain that we saw. That part is hydrophobic. So here again, you can see why it's referred to as a fatty acid, because you can see that hydrophilic end is a carboxylic acid group, and the hydrophobic part is a non-polar tail, which is an aliphatic hydrocarbon chain. So hydrophobic is a fat. The fat is insoluble in water. So you have a fatty acid structure of that nature, which we say is amphipathic. Now, we can look much more at this, hydrophobicity of fatty acids much more practically, that that when you pour oil into water, the oil does not mix with water. You see that in this, diagram here, you have oil, which is floating on the surface of the water. Now, you can see clearly that the oil that is floating on the water has the polar ends, that is the carboxylic acid ends, or say the hydrophilic ends, pointing towards the water surface, whilst you have the hydrophobic, hydrocarbon chain pointing upwards. Because of course, we have just seen that the carboxylic end is hydrophilic, and so that can have interactions with water molecules, whilst the long chain hydrocarbon is hydrophobic, and so those will turn away from the water molecule. So that's why you see such floating of oil on water. But apart from that, if you should shake that mixture, say the oil with water, you can have some uh, uh, structures forming also in that mixture, and you can have what is being referred to as a micelle, where you have, say, the hydrophilic ends pointing outwards, having interaction with water molecules, whilst the hydrophobic ends are pointing inwards. We'll see it much more in detail. And you can have some bilayer structure also forming. This bilayer vesicle also forming in such a mixture. If you should shake an oil with water, some droplets do form, and this is what can happen. You can have either some single layer that way, or you can have some bilayer in that manner. Now, these lipid bilayers that can form, you do see that the hydrophilic ends, that is the carboxylic ends, that's an acid that can get protonated, deprotonated. And so those parts, the hydrophilic ends, do carry some charges. So you can have, say, charged surface of such bilayer that forms. So you can see, this bilayer being shown here, that you have charged surfaces and you have the hydrophobic tails. I mean, those are the hydrocarbon chains that are pointing towards, each other. I mean, it's just like, look at it this way, that water will mix in water, oil will mix in oil. So hydrophilic ends will point towards the water molecule, whilst the hydrophobic ends will point towards each other. Now, you can also have structures of, a more complex nature, that are forming from these bilayers, so that you can have, a bilayer, structure, the vesicle which we saw. If you look at it in 3D form, this is how it will look like some basket that is forming. And inside it, because you have the hydrophilic ends pointing outwards, you can have water filling in there. And of course, such structure can also have, water molecules around it that it will be interacting with. In fact, when we come to see the structure of the cell, then you realize that actually you can refer to the cell as some, a lipid, vesicle of the salt that is having all those organelles inside of it. And so you see that the hydrophobic tails are pointing towards each other, and you have the hydrophilic ends pointing outwards that can have interaction with an aqueous environment. Now, you can have what is being referred to as liposome. What we saw much earlier, we are now seeing it in much more detail, because in that water mixture, it was just drawn, just a cross-sectional section. But now you can see here that the liposome is more like a spherical structure that you can have some aqueous environment freely in it, and you can have aqueous environment around it. Actually, you've seen some liposomes before. If you see, there are some types of food supplements that are like capsules, but like some soft capsules. These are liposomes. Okay, I see Eugene Mensah is saying that he cannot see my screen. Eugene, if you can't see my screen, I guess that the problem is from you. Usually, your friends do advise go out of the class and come back in, and perhaps it will come. It's possible you have some network issues, some technical issues there, but the screen is showing. All the others can see it. All right. Or you have a micelle. The micelle, you can see the micelle is a single layer structure, so that it's only the outside of it that can have interaction with an aqueous environment. Usually, some of these micelles, they are some form of transport, lipid transport structures. So in lipid metabolism, you can have, structures like this that can have, lots of lipids carried, that they can travel, get transported in the aqueous environment of an organism. Because once you have, this micelle, which can have, lipids, or say hydrophobic molecules that are packed inside here, then that micelle can easily travel through an aqueous environment because it's having just the external part, which is hydrophilic, having interaction with the aqueous environment. So that can get transported easily. And as I said earlier, you come to see that if you look at the cell, so what we refer to, say, the cell membrane, for instance. So the cell membrane will look like this, like the liposome that we are looking at. That you have inside of it all these organelles, like the nucleus, endoplasmic reticulum. You can mention them. All of those organelles which are found in cells are sort of, say you have such a big, like a liposomal structure in which you have these organelles inside there with an aqueous environment, what you are calling the cytoplasm inside there. And so you see the cell can easily interact with the aqueous environment outside of it. And inside of it is also an aqueous environment. And it can get protected well with that lipid bilayer. Right. Now you want to remind yourself of some of these fundamental things about the structure of fatty acids. And you do remember you have some different types of fatty acids. And one of them is a saturated fatty acid. And what's a saturated fatty acid? A fatty acid that has only single carbon-carbon bonds in them. And so here you see there are no double bonds here. Every bond between, carbons, you have seen these structures before, so I think you understand. From first year, you would have studied how to represent fatty acids, just by using these skeletal structures, where the corners of these structures represent carbon atoms with hydrogens on them, and you can show the carboxylic acid group, which is at the end of the structure. So those are saturated fatty acids containing only single carbon-carbon bonds. You do also have unsaturated fatty acids. And these unsaturated fatty acids, they contain one or more double bonds. And you can have what is referred to as monounsaturated fatty acids. These are fatty acids with only one double bond in them. Like you see here, oleic acid is having only one double bond in it. Polyunsaturated fatty acids will have more than one double bond in them. So for instance, you have linoleic acid, which is having two double bonds in there. So that's a polyunsaturated fatty acid. Now, you also want to remind yourself of the numbering of the carbons in the fatty acids. And so we see the structure above. The carbons are numbered from 1 up to 18. And take note that the numbering of carbons in fatty acids always starts from, if you are using these numeric carbons, the numeric numbers 1 to 1, 2, 3, 4, 5, the numbering starts from the carbon that is carrying the carboxylic functional group. So the carbon of the carboxylic functional group is always a carbon number 1. So it doesn't matter how you write this structure. You could write this structure, turn it, flip it to the right so that the carboxylic acid group is on the right. It means that if you are doing that numbering, you have to start numbering from the right to the left. But there are other forms of numbering where you have the carbon which binds to the carboxylic acid group carbon is the alpha carbon. So that carbon, the carbon number 2, is the alpha carbon. The next to it, the beta carbon. So this is, using some Greek alphabets also in numbering. You can have the alpha, beta, gamma, epsilon, etc. But of course, in lipid metabolism, the most relevant of these, numbering is the alpha and the beta. Usually, that's what is referred to, because in lipid metabolism, we do refer to what is referred to as alpha oxidation, beta oxidation. So you do have that. Now, you also have what is referred to as, this numbering from the end of the fatty acid. That is not the carboxylic acid end, but the other end. And that is numbering with omega. Omega also, a Greek letter meaning the last. So the last carbon is omega 1. And you have omega 2, omega 3, like that. And I think you came across also the fact that it is this kind of numbering which is used referring to some specific fatty acids, as say omega 3, omega 6, omega 9, which is just to say that these fatty acids have double bonds at the ending of the fatty acid. To say that these fatty acids could easily break down in metabolism. That's why they are being referred to as some. Usually, some of these are some essential fatty acids, and because of these double bonds they have in them, they can easily be broken down, and so they are. Some foods are marketed as such that they contain some of these omega 3, omega 6, omega 9, or even some food supplements are marketed that way, that they have this content in them, meaning that they have these essential fatty acids, or they have these fatty acids that can easily be broken down, and so they are healthy. That's all it is that is in these marketing messages. Now, you also have some numeric representation of these fatty acids, which also you want to revise yourselves. You want that you can represent a long chain fatty acid just with numbers, And so, for instance, if you take a look at this structure, it's having 18 carbons in it. And so you see the numbering from 1 to 18. And so you see the first number there is 18, which is referring to the number of carbons. And you have 18:1. What does the 1 there mean? The 1 means the number of double bonds that are present in the fatty acid. And you have delta 9. Delta 9 is telling us the position of the double bond. So this whole structure over here can be represented as 18:1 delta 9. And delta 9 means the double bond is on carbon number 9. That is, if you are counting from 1 up to the last number, the double bond between carbon number 9 and carbon number 10. That is what is represented as delta 9. Take note that this is not the same as having the double bond between carbon number 8 and carbon number 9. A lot of students make such mistakes. Maybe I can give you some of the favorite questions I give are just to give you this. For instance, the numeric representation of the fatty acid, like 18:1 delta 9, and tell you to give me the structure of the fatty acid. And you see some students will draw the structure and then they put the bond between carbon number 8 and carbon number 9 instead of the bond being between carbon number 9 and carbon number 10. So you want to pay attention to that. If you do that, you lose marks. It's wrong. It's not the same as the 18: delta 9. All right, now still on this numeric representation of the fatty acids, what the example we just saw was showing, double bond in a fatty acid, but that double bond was only 1. How about if you have more than 1 double bonds? How do you represent that? So for instance, this fatty acid, linoleic acid, is having the numeric formula 18:2 delta 9 12. So 9, 12. Because you can see clearly that it's having 2 double bonds in it. First double bond is on carbon number 9, and the second double bond is on carbon number 12. That's what it means. So if you have more than 1 double bonds, you can have just the numbers written with comma between them. So you can have, for instance, 18:2 delta 9 12 15. So 9, 12, 15. So that will be 18:3 delta 9, 12, 15, telling you that the fatty acid contains 18 carbons, 3 double bonds, and then the location of the double bonds are carbon number 9, carbon number 12, carbon number 15. So you can have anything like that, and you should be able to quickly represent it in such a structure. Those are some very fundamental, simple questions that I put to students to, see your foundation in this, because this is very, very foundational, that even if you are sleeping, you are called and given the number, you should be able to quickly give the structure of the fatty acid without thinking about it. All right, now we want to pay attention also to the dependence of the melting point of fatty acids depending on the number of carbons that are present in the fatty acid, as well as the number of double bonds that are present in the fatty acid. How do these affect the melting point of the fatty acid? Now, these you come to see, these are important because when you take, say, the structure of biomembranes, which you see that they mostly consist of, some phospholipids, which of course will consist mainly of these fatty acids that are bonded on some backbone. Depending on the type of fatty acids that are present in the lipid, the biomembrane, you have, that biomembrane exhibiting a certain property, whether it is going to be more fluid or that membrane is going to be less fluid, or how it's going to be, withstand temperature. You remember we did talk about, say, some thermophilic bacteria, for instance, that can survive temperatures even above 100 degrees, So why that? I mean, you can explain it with some of these, the type of fatty acids that are present in the membranes of these organisms. All right, Prince Okine is saying some people are not able to join. He keeps saying call ended. Well, I don't know how to help in such a case, because as you can see, we are here. We have not ended the call. We are still on it. I think it's such a network problem for some people to join, but I hope that as I speak, all of you who are online, you can hear me clearly and you can see my screen. So whoever is not able to join, obviously the problem is coming from their end. There are technical issues which they will have to sort out themselves. All right, so if we take some fatty acids at the top end here, we have some fatty acids which are saturated fatty acids, meaning they don't contain any double bond in them. So we want to see, if you look at a melting point, what trend can we see for the fatty acids, so that we can deduce how the number of carbons in the fatty acids can affect their melting point. Now, if you pay attention to the number of carbons, under the column there, which is abbreviation, you have the number of carbons starting from 10. So 10, 12, 14, 16, 18, 20, 22, 24, 26. And you can see all the saturated fatty acids are having 10, the colon, after the colon, 0, 0, 0, 0, meaning they don't have any double bonds in them. So the number of carbons increase downwards from 10 up to 26 carbons. Now let's come to the last column, melting point, and see what do we notice? We notice that melting point starts from very good. So Raboni, Adobia Osai says melting point increases as the number of carbon atoms increases. So the last part is not that they are inversely proportional. They are directly proportional, because both sides, if you take the number of carbons, there's an increase. And when you take also the melting point, the melting point is also increasing. Yes. So yes, Adobia, yes. So directly proportional. Melting point increases with increase in number of carbons. Good. How about if you come to the unsaturated fatty acids? But of course, logically, why that? You see, when you have number of carbons increasing, it means you are having the size of the molecule increasing, which means that more energy will be required to break down a bigger molecule. than a smaller molecule. So that is a very, coming from a very logical reasoning, you can understand this. So that even if you go running some chemical experiments in a laboratory, you will see this result, all right. But logically, it makes sense that yes, once you have the number of carbons increasing, you are having the molecule becoming bigger, and so you need more energy to break down such molecule. Now, if we come to the unsaturated fatty acids, starting from, the first one, 16:1 delta 9. So that is having 16 carbons, one double bond, which is on carbon number 9. Then the next unsaturated fatty acid is having 18 carbons, one double bond, and that double bond is also on carbon number 9. But I say here, if you compare those two, you realize that melting point increases from 0 to 16. Now, melting point increases because, as you can see, the number of double bonds are the same, 1, in both of them. But there is an increase in the number of carbons. So just like we were looking at the saturated fatty acids, even though you take the unsaturated fatty acids, you see that the number of double bonds is constant in these two. It is the number of carbons that increase. And so you see that there is an increase in the melting point as well, from 0 to 16. But let's come down to the next, where now if you take that second fatty acid, which is containing 18 carbons, one double bond, and you compare it to the fatty acid below it, linoleic acid, which contains 18 carbons. So the number of carbons is constant, the same, but you have the number of double bonds increasing. So you have 2 double bonds in that, which those double bonds are on carbon number 9 and carbon number 12. You realize that there is a decrease in the melting point from 16 to 5. So as the number of double bonds increase, it results into a decrease in the melting point. So this is an inverse proportion. So these two, logically, if you look at it, if you have a structure that contains 18 carbons, let's say the structure is having the same molecular weight, and yet you are having 2 double bonds in one of them. What does it mean? A double bond is weaker than a single bond. And so lesser amount of energy will be needed to break down the structure that has more double bonds in it. And so that's why you notice that as the number of double bonds increase, the melting point decreases. And if you come again to the octadecanoic acid, linolenic acid, where now the double bond increases again, so the number of carbons still remains constant, 18, but you now have 3 double bonds in that structure. You notice that the melting point decreases much more, from 5 to -11. You know what that means? It means this oil, the linolenic acid, at -11 degrees Celsius will still be liquid. That's the meaning. -11. That's a temperature much below the temperature that you have in your refrigerator, which is usually, the refrigerator you use at home, which is usually like say -5, and you have this -11, and that oil will still be remaining liquid. And you can see a fatty acid, arachidonic acid, even though you have the number of carbons increased to 20, it is having 4 double bonds in it. And because of those 4 double bonds, you see that the temperature goes all the way to negative 50 degrees Celsius, melting point. I mean, these things are applicable even in, things like antifreezes. You have some products, the antifreezes, that usually are used in the temperate regions where we have winter, that uh, when you wake up in the morning, you see after it has snowed and all of that, you have ice covering all your windscreens and things like that. You can use these antifreezes, spray on the screens, and then you'll be able to clean your windscreens, things like that. Or in the development of uh, say engine oils, for instance, you want to have oils that can withstand very low temperatures, that even when the temperature is so low, the oil will still be liquid, that you can still run your engines. So these are very practical things, that they are not just some abstract things that you are studying. All right, so we've seen that relationship between number of double bonds with melting points. We've seen the relationship also between the number of carbons with melting points. Now, I mean, you come to see that in membranes, those things are important, because depending on the type of fatty acid that is present in the membrane, you can tell some physical chemical properties of such a membrane. That's why you need to understand these fundamental things to be able to understand, for instance, why a particular bacteria can be thermophilic but the other cannot. Why some cell can withstand a certain condition but another cannot. Things like that. You also want to remind yourself of, these cis and trans configurations. You did these things even in secondary school, where you had these alkenes, that when you have the bigger residues at one end of the double bond, you have an increase in what is referred to as a steric repulsion. I mean, that's just like some interaction between, the atoms that are within a molecule. So that you can see that hydrogen is the smallest atoms. They are at one side, whilst you have, more bulky part of the molecules at one side. Of course, this will be having some kind of repulsion between them. And you have, the trans configuration, where you have the bulky portions of the molecule at opposite sides, and then you have the small hydrogen atoms also on opposite sides. Of course, this one you have less steric repulsion, because of course the bulky sides are not at one side to be having such a repulsion between them. And what does this mean for fatty acids? Now you can have fatty acids in their cis or trans forms. I mean, the unsaturated fatty acids, you can have them in their cis or trans forms. But of course, naturally, say if you take the cell, let's say cell membrane, or say you take biological systems, say even the oils, say plant oils that are extracted from plants. Let's say your cooking oil, for instance. Now, these fatty acids that have the unsaturated fatty acids that are present in those oils are in their cis forms. They are naturally in their cis forms, which means that, you see, we were talking about steric repulsion. And because of such steric repulsion, you will have in the fatty acid some bend, say a kink in the structure of the fatty acid, because you're having the bulky sides at one side and the hydrogen, the small atom also on one side. You have a certain bend in the molecule. And that bend alone also, results into a weakening of, the bond the more that the molecule is more unstable. So it can break down, much, much easier because of that bend in the molecule in its cis configuration. But you do have the trans configuration is a more stable form, because you can see it's more or less like a straight structure. So that bond will be more stronger than the bond that is having a bend in it. Now, health-wise, if you want to apply this practically, you do hear the advice from health professionals that, like, for instance, your cooking oils, you shouldn't be using them over and over again. You use it preferably only once. That is because cooking oils have this cis configuration in them, that each time, but you see, there cannot be rotation around that double bond, so that you say that cis can easily change into trans. No, before a cis configuration converts to a trans configuration, there must be a breakage of the bond and a reformation of the bond before you can have such trans configurations occurring. And this happens when oil, for instance, is being used to cook. The heat breaks those bonds, and if you cool the oil, there can be reformation, and you have such trans configurations of the oil forming. And so the more repeatedly you make use of the oil, the more the trans configurations of the oils are formed, and which makes it more unhealthy. So the oil will now be having more trans configurations in them, which will require more energy to be broken. That's why it's advised that cooking oils are used only once. All right, so Osai says I should explain the bend and the breakage again. So I was talking of the fact that you have a cis configuration that is having the bulky part of the molecules at one side of the double bond, whilst you have the small hydrogen bond, the small hydrogen atoms also on one side. And because of that, because you have a repulsion between these bulky groups, you have more steric repulsion between the groups, which results into a bend in the double bond. And if you do have the bulky parts on opposite sides, that's almost, less of a repulsion between the bulky groups, and so you have almost a straight chain structure that forms instead of having such a bend. So the cis configuration has that bend, but the trans configuration does not have that bend. And because of the bend, it's sort of a stretch that is on that bond, and so the bond is weaker in the cis configuration. All right. Now, you see that also, such bonds also in fatty acids, which will be present in, say, membranes, will result into either a particular portion of a cell membrane will be easily penetrated, can be easily penetrated by substances moving in and out of the cell. Like you have portions of the cell that are like pores within which substances can pass through and go out, and some places are, are much more, dense and stable that substances cannot easily pass through there, depending on the type of fatty acids that are present at a particular region of fatty of a membrane. You have a particular function of transport that can occur there or not. All right, so that's what I was talking about. Stability, where you can have stable and some unstable fatty acid aggregates. So for instance, if you have this this is depicting some aggregates, fatty acid aggregates with only saturated fatty acids. And you can see that the hydrophobic tails are all straight. So this is actually consisting of saturated fatty acids, no double bonds in them. And you realize that the aggregate which is formed here is very tightly packed together, and you don't have any sort of instability in there. But if you take the aggregate which is on the right hand side, which is having a mixture of saturated and unsaturated fatty acids, such that you have the unsaturated fatty acids having bends in them. I mean, those bends that we just saw in those fatty acids in the cis configuration, then you realize that this aggregate is unstable, because you can see there are spaces, much more spaces in between these fatty acids. So you can have such unstable fatty acid aggregates as well present in membranes. Now, you do also remember that, say, the storage form of fatty acids, I mean, are the triacyl glycerols. I mean, it's just like if you take carbohydrates, for instance, you have the storage form of these carbohydrates being starch in plants, where you have lots of these glucose molecules binding together. Between them you have these glycosidic bonds, and you have these glucose molecules packed together, forming this structure of starch. I mean, similarly, when you come to say animals, let's say human beings, for instance, we also do store starch, say, in the liver and in the skeletal muscle in the form of glycogen. And these are also, it's just glucose molecules that are packed together so that later, particularly in the night when you are not eating, you are sleeping, then the body will be making use of that carbohydrate which is packed in the glycogen in the liver. Or maybe if you are involved in some vigorous activity, the glycogen which is stored in the skeletal muscles will be breaking down to be generating energy quicker for the muscles, for the muscle cells. The same way you have these fatty acids that can get packed onto a glycerol backbone. So it's a glycerol. A glycerol is having 3 carbons with hydroxyl groups on them. And you only see that in place of those hydroxyl groups, you have a esterification of, say, 3 fatty acids. That's why we say triacyl glycerol. So 3 fatty acids then get esterified on the glycerol backbone to give you a triacyl glycerol. So that is packing of 3 fatty acids together on one backbone. And the backbone here is glycerol. So which is why you do here when it's a mantra from the secondary school or even from the junior high. You do mention that fats and oils, break down into fatty acids and glycerol. I mean, that's what it means. Lipids, when they get metabolized, they break down into fatty acids, which is, say, these fatty acids get hydrolyzed from the glycerol backbone. And so you are left with the free fatty acids and then a glycerol. That's what it means. Okay, now, if you look at it much more in detail, so that's the glycerol in the blue, triangle, the blue box. The glycerol is the backbone onto which you have these three fatty acids forming the bond which is formed between the fatty acids and the glycerol backbone. These are the ester bonds. So the esters, the esters have this structure, a carbon bonded onto oxygen, which is bonded onto a carbon, and that second carbon is having a double bond, oxygen, on it. So that's an ester. That's why we say that the fatty acids are esterified onto the glycerol backbone. And the reverse of that is de-esterification, removal of the fatty acid from the glycerol backbone. So, I mean, if you are talking of it in terms of metabolism, that process of hydrolysis of fatty acid from the glycerol backbone is de-esterification. I think in metabolic terms also is referred to as a lipolysis, removal of the fatty acid from the glycerol backbone. All right, so that is to understand, how these structures are. I mean, whereas some of these very simple, interesting questions I give in this aspect of things is to give you three fatty acids. I can give you the fatty acid in terms of its numeric formula. Let's say I say that you have three fatty acids, 16 colon 0, 18 colon 1, delta 9, 20 colon 2, delta 9, comma 12. I give you these three fatty acids, and I say that you should use these three fatty acids to form a triacylglycerol. And which is also, those are simple things to do that you should be able to quickly have that glycerol backbone and, have the esterification of those fatty acids onto the glycerol backbone to form the triacylglycerol. And some of these questions are simple that, I shouldn't have students getting them wrong. Okay, so Joyce Thompson says people are waiting to be added to the meeting. I don't have to add anybody to the meeting. People can everybody can get into the meeting without anything. You were able to get into the meeting without any problem. So why should some people not be able to enter? I don't know what it is with them. All right. They just have to tap on the link and join. The link is not restricted to anyone. Anybody can tap and join it. Okay, so we can continue and look at some other type of these lipids that you can come across. It's waxes. What are they? So yes, the waxes are also esters, and these are esters of fatty acids, and fatty alcohols. So, fatty alcohols are, usually alcohols of a higher number of carbons. Usually, fatty acids are counted as, say, carbon number 4. That's like butanol, all the way up to, say, the carbon number 26 and more. Now, but for waxes, these are formed between fatty acids consisting of, say, 14 carbons up to, say, 36 carbons. Then you have them getting esterified with fatty alcohols that contain 16 carbons up to, say, 30 carbons. And you see, we just talked about melting point in relation to the number of carbons. And if you take a look at this number of carbons that you are seeing here, these are a high number of carbons in there. So that can tell you why you have, wax, say the bee wax, having such a semi-solid, structure. I mean, if you look at the bee wax, you see that it's semi-solid because, as you can see, the number of carbons in there is so high, and so the melting point will be high. And so you can see, for instance, the bee wax is if you look at the structure above here, the bee wax is being formed between palmitic acid, which consists of 16 carbons. So this part is palmitic acid consisting of, say, 16 carbons. And you have triacontanol that consists of 30 carbons. So these two, when they get esterified, of course, that will be resulting into such a structure, the bee wax. So see, you can have palmitic acid, triacontanol, and even synthesize, do some esterification synthetically, which is why you do have some products that are made of bee wax for polishing, maybe in some candles. You have even polishes for say fruits. I mean, some fruits are sort of coated with bee wax. Some wax, and that will make the food look nice, shiny in the shops. So you like to buy them. If you go to the automobile industry, they do use bee wax to do polishing of the vehicles when they are putting them in the showrooms. You have polishing of these vehicles with waxes. It will shine before, so to attract you to buy them. Similarly, waxes are used even in the furniture industry, for instance, where they are used to do polishing of furniture. And also, usually when they are in the showrooms, they do polish them with these waxes. Even your shoe polishes contain some wax in them. All right. But Prince is saying that Prince Okine says it keeps saying call end. Well, I don't know what it is with you. I think you are just having some network problems, really. But I don't think you should worry so much because we are having a recording of this lecture. And as I always do, after the lecture, I share the video with you. So you can listen fully to the video. Even the video, I think you can easily download it, and then you can watch continuously without any breaking in it, because these recordings are happening, very well. I've monitored it and I've seen that the recording does not have any breakages in them, so you shouldn't have any problem anyway. So don't worry so much about if you are experiencing any breakages. I think the videos will help you later when you are doing your revisions. You can hear everything well together with the presentation slides that I'll share with you. You can make use of them together, and then everything will be okay. Now let's move on much more specifically to membrane lipids. Because the course is on membrane biomembranes and enzymology, and we finished the enzymology part, and we are now moving on to the biomembrane part. So we move specifically to the membrane lipids. Now, Samuel Latte says the more the number of carbons determines its melting point number. And then he says the waxes. Yes. So you mean for the waxes? Yes, of course. Just like we saw with the lipids, the fatty acids. Just like we saw with the fatty acids, that the melting point increases with the number of carbons. It also means that for the waxes, depending on the fatty acid and the alcohol, which are components of the wax, you can have melting points that will be lower or melting point that is higher. So the question you are asking is, let's say, for instance, we take a fatty acid that contains 14 carbons, and then we take a fatty alcohol that contains 20 carbons, and compare that to wax that contains a fatty acid of, say, 30 carbons and a fatty alcohol that contains also 30 carbons. Of course, the second wax is going to have a higher melting point than the first. So yes, the more the number of carbons will determine, or the more the number of carbons of the alcohol and the fatty acid, which are components of the wax, the higher the melting point of that wax. Yes, Samuel, you are right. All right. I like the way you are connecting, these thoughts and deriving these things out of it. I mean, it tells me that yes, you have an understanding of these things, which is good. You have the membrane lipids. We can divide them into different types. You have the phospholipids, glycolipids, and what we refer to as archibacterial ether lipids. Now, under the phospholipids, you have glycerophospholipids and sphingolipids. And you see the glycerophospholipids are having a glycerol backbone. Then you have two fatty acids that are bonded on that glycerol. Then there's a phosphate group. So you see why I say a glycerophospholipid. You have a glycerol backbone, you have a phosphate group, and on that phosphate group, you can have a side chain that binds there. Not necessarily an alcohol. It can be anything. It can be a fatty acid. Even sometimes it can even be an amino acid in some structures. If it's some lipoproteins, for instance, you can have such complex of structures forming. Then you have sphingolipids. So for the sphingolipids, you see it's having a backbone, which is not glycerol, but is sphingosine. So that's another lipid. We'll see the structure of sphingosine. And on that sphingosine, you have a fatty acid. Again, you have a phosphate group. So you see why is sometimes is being referred to as a sphingophospho. lipid. But here it's written as sphingolipid. But of course, it is under phospholipids. And so you can say, even though it says sphingolipids, you can say sphingophospholipid. And then on that phosphate group also, there's a side chain. Here they've written, say, a choline, but of course, that side chain on that phosphate group can be any residue there. Then you have glycolipids. So from the name glyco, then you know this will have some carbohydrate content. So you have here too, there are some sphingolipids because the backbone is sphingosine, and you have on the sphingosine a fatty acid, and then you can have some monosaccharide or some oligosaccharide binding on there. And so then you have such a you can say that's a sphingoglycolipid. Then you have galactolipids, which are also referred to as sulfolipids, because in some of them you do have some sulfate groups. You come across this more in plants, and you have the glycerol backbone, and you have fatty acids on that glycerol backbone. Then you have the carbohydrate, which can also be a monosaccharide or disaccharide, and you can have some sulfate group or attached on those monosaccharide or disaccharides to give you sulfolipids. Then the archibacterial, the actibacterial ether lipids. These are more complex membrane lipids. And as you can see, because of that such a complexity of the structure, obviously, the membrane which will be formed by such lipids will be very tough. I mean, you'll find this in what we do refer to as the thermophilic bacteria, because this will be much, much harder to break down because of the complexity of the structure. You can see you have 3 glycerol backbones. And between them, you have even more complex fatty acids, branched hydrocarbons, diphatanyl, and you have phosphate group that is, binding the second glycerol and that last glycerol backbones together. And we'll come to see why we say ether. You can pay attention to that linkage there, which is shown with the red lines. So it's an ether linkage. And pay attention that these are not esters, they are ethers. So we see the difference between ester and ether. So what we'll do is that we'll take each of these. Like we'll take the glycerophospholipids, look at them, then we'll move on and go to the sphingolipids, look at them, then we move and look at the galactolipids, and then lastly, we look at the archibacterial ether lipids. And I think for today, we'll end there and continue the next time with other things. So let's start to look at the glycerophospholipid. Take the glycerophospholipid structure. And we see the hydrophilic head of the glycerophospholipid structure consists of that the glycerol backbone, which is shown there. You have the 3 carbons, which originally should be having hydroxyl groups on them. But on this glycerol backbone, you see you have a phosphate group, which is also highlighted in that yellow region, the phosphate group. And on that phosphate group, you saw that there could be a side chain that is bound on it. And that side chain can be carrying a negative charge. So why are all these people writing I in the group? So you have this phosphate group which is charged, and you have you have these fatty acid side chains. You have these fatty acid that are bonded on, the two carbons there. So these are the hydrophobic tails. I mean, if you are looking at the structural formula of this, glycerophospholipid in detail, this is what you see, that you have the hydrophobic tails, which are the fatty acids, and you have the hydrophilic head, which consists of the glycerol with the phosphate group on it. Of course, you'll see some computational, structure, some modeling of such, to show the phosphate group, the fatty acid chains. But this is just, some kind of modeling. You don't see the full structure in detail as we have seen in the first part. And you also, when you are writing, a lot of times you just do, maybe you draw a circle and you do 2 lines under it to show that this is a phospholipid, or even sometimes it's just to show that is some glycerol-lipid, like you have a glycerol with some fatty acids on it, you have a side chain like that. So you do see representation of these things in writing, like a circle with some two legs under it. So you can say that. All right. So that's the structure of the glycerophospholipid in detail. Now, if you look at the structure of the different glycerophospholipids, what we have just looked at, you have the glycerol backbone, and you have 2 fatty acids that are esterified on the glycerol backbone, and you have a phosphate group on that last glycerol carbon, and the X becomes the head group substituent. What we say that that can be any other component that binds there. So for instance, examples that we see here, if that X is just hydrogen, it means you just have a phosphate group there. That is phosphatidic acid. And phosphatidic acid is actually the parent molecule of all glycerophospholipids, because that's just a phosphate group, and it is on a phosphate group that all the other substituents come to bind. And so you can have these lipid membranes being things like phosphatidyl ethanol amine, and that you have ethanol amine will come in place of that X as a substituent. Similarly, you can have choline, which you find in phosphatidyl choline, and you see serine. So serine is what? It's an amino acid. So you see, I was mentioning that you can even have some amino acid binding there when you're having some maybe phospholipids, in complex with some proteins. You can have such. So that would be like some lipoproteins. Now you also will see even in place of such, the substituent, the X, you can even have another molecule of glycerol. So you have a phosphatidyl glycerol. And then you do have, some other structures like inositol 4,5-biphosphates or phosphatidyl glycerol. So you see another phosphatidyl glycerol that comes to bind on there. So that's another complex one, which you find in say cardiolipine. Now all these structures, you come across them carrying out various functions in the cell. For instance, You have choline, let's say phosphatidylcholine, phosphatidylethanolamine acting as some neurotransmitters. So you find them more in this, in the nervous system. You find these things in different cells, which will be carrying different functions in the cell membrane. We'll see more of this maybe for cell recognition and different things. Similarly, you have the sphingolipids. So we move on to the next, which is the sphingolipids and the glycosphingolipids. I mean, that is to say that in the glycosphingolipids, you have some carbohydrate there, some of those ones that were having some monosaccharides binding on there, or even oligosaccharides binding. Those will be like glycosphingolipids. So we see the sphingosine, which is a 16 carbon structure, which is shaded in pink, so you can relate with it. And on that sphingosine, which is the backbone, you have a fatty acid. And then on that, first carbon there, you have a head substituent. And the head substituent, if it is hydrogen, you have that to be ceramide. So again, ceramide is the parent molecule of all sphingolipids. So you see, in place of that, hydrogen, if you are having a phosphocholine, you have what is referred to as a sphingomyelin. And the sphingomyelin is also one of these, lipids you find in nerve cells. If you remember, I think you were in secondary school, you drew a nerve cell and you were showing what's referred to as a myelin sheath, protecting the axon of the nerve cell. So that is component of those nerve cells, the sphingomyelin. And you see, even in place of that X substituent, you can have glucose, and that is glucosyl cerebroside. So again, you find this in the nervous system, because the name even tells you cerebroside. So this will be in the neurons, which will be carrying out functions of nerve signals. You have the lactosylceramide, which sometimes also be referred to as a globoside. You can have in place of that X, the head substituent, having some disaccharide, trisaccharide, tetrasaccharide, so like some oligosaccharides that are there, as the head substituents. These you find them also in the nervous system, which is why you notice that some of these neurodegenerative disorders are related to some of these lipids. Some deficiency in their metabolism results into some mental retardation and some other neurodegenerative disorders. Ganglioside, what is referred to as ganglioside GM2, is having that head substituent also being this oligosaccharide, which is consisting of glucose, galactose, some 5-acetyl-neuraminic acid, N-acetyl-galactosamine. These that structure of some oligosaccharide that is composed of not only the monosaccharides, say glucose, galactose, but also with some derivatives of these monosaccharides. And all of these are going to be in the cell membrane. I think one of the things that you have to understand is, so for instance, when you do some extractions of oils, I think we'll come to it. Let's move on. We'll come to, we'll speak briefly about it at the end of it. So you also have, some of these sphingolipids that are involved in biological recognition. So, for instance, we do have, say, the blood grouping of, say, the ABO blood group. You have blood group A, blood group B, blood group O, blood group AB. What do these mean? In different individuals, you have this, the cell that so these immune cells that's in the membrane is having some specific lipids that, if you, for persons that are, say, blood group O, they have what's referred to as the O antigen, and that O antigen, consists of this, contains this lipid that the side chain, that is found, or say the head substituent on that sphingosine, contains this oligosaccharide where you have glucose, galactose, N-acetyl galactosamine, galactose, and fructose. Now, what you notice is that for the O antigen, that oligosaccharide that is in the O antigen is constant in all the other, blood groups, except that for the A antigen, there is also, an extra N-acetyl galactosamine, which is bound on that. Then in persons with, say, B antigen, they have galactose bound onto that oligosaccharide, which is present for the O group. And so what it means, persons that are blood group AB will contain both A and B antigen. So you do remember these things. So this the foundation for it that you have in different persons, these different glycosphingolipids, which are determinants of the blood groups based on this ABO blood group classification. All right. Now the galactolipids. So I mentioned that these galactolipids, you find them in plants. Usually you find them in the chloroplasts, in the organelle responsible for the photosynthetic process in plants. And these are lipids. So this is why I was mentioning earlier that if you are doing some extraction from plants, say you are doing some oil extraction, where exactly are you extracting the oil from? If you think of it, where exactly are you extracting the oil from? I mean, you are extracting the oil from the cell membranes, because it's in the membranes that you have a lot of these lipids. Because you have the membrane is mostly a lipid bilayer in which you have some other components that are embedded. When we come to see the structure of the biomembrane, then you will see that yes, the biomembrane is just completely a lipid. So that the extraction that you do, whether extraction of, say, maybe cooking oil from, say, granules, or anything, maybe coconut or anything like that, the extraction which you are doing is actually coming from the membranes of the cells. of these plants. So you have this, you can have the monogalactosyl diacylglycerol. So you see the glycerol backbone, and on the glycerol backbone, you have two fatty acids esterified, and then that last carbon there, you have galactose, one molecule of galactose there. That's a monogalactosyl diacylglycerol. You can have even a digalactosyl diacylglycerol, which is to say that you are now having a disaccharide of two galactose residues. So you do see these structures in plants. And you come across what is referred to as the eta lipids. So we are moving to the last end, where we are talking about the last lipids, which I mentioned you should pay attention to that term eta, because an eta is different from an ester. So even if you take a look at these two, you see the bond there, which is shaded in red or in pink. You see the bond which is between the oxygen of that, the hydroxyl group that came from the glycerol backbone. You see on the carbon that binds onto it, there's no double bond O there. So an eta bond is COC, carbon, oxygen, carbon. That's an eta. But you see earlier on, we saw the structure of the ester, which is the second one which you are seeing here, which is COC, and on that C, there's a double bond O. That is an ester. So that's the difference. The bond you see on carbon number 2 is an ester bond, but the bond you see on carbon number 1 is an eta bond. So you have COC, eta, and you have COC, double bond O, ester. So you notice the two differences between an eta and an ester. Then, of course, the example we are seeing here is a plasmalogen, where you have a phosphate group, and then on the phosphate group, you have choline. And these plasmalogens also, they carry out different functions in the circulatory system, in the nervous system, in the immune system. You find these structures, the plasmalogens. You have what's referred to as the platelet activating factor, which has a major role to play in inflammatory reactions. And if you look at the structure here too, you can see that on carbon number 1, you have an eta linkage. Well, here it is written very well, so you can see the difference between the eta and the ester, because it's written at the top there for that first bond, which is shaded pink, eta linked alkane, and then down here, the second bond is written clearly acetyl ester. So you can now see clearly the difference between an eta bond and an ester bond. Okay, somebody say you can't hear anything. Joyce Thompson says she can't hear anything. Well, I don't know. I think the others can hear. Maybe the problem is from you. But like I said much earlier, if you are having such problems, don't worry so much, because later when you have the video, you can watch it fully and you hear everything, because the recording is good. I see the number has reduced. Some people have left the class. All right. Anyway, it's all being recorded. Who came at what time and who left at what time. Now, we have the platelet activating factor, also having choline there, on that phosphate group. And I was mentioning that this, the platelet activating factor has a major role to play in inflammatory reactions. I mean, just the name, platelet activating. So this compound that activates platelets. So let's say if there's some inflammation, this factor, this compound activates, is released by, say, red blood cells, and this activates platelets, and these platelets then trigger various cascade of reactions, which result into, say, formation of what we refer to as fibrin. It's some fibrous protein structure from its precursor, fibrinogen, and that fibrin will form some mesh which will trap red blood cells. So, for instance, if there's a cut, in order to stop the blood from bleeding, to stop bleeding, this is the reaction, a cascade of reactions which occurs. A mesh is formed from fibrin that will trap some red blood cells, and over time there will be some thick covering which is formed, and that is going to stop the bleeding. So that's this platelet activating factor, that's a major function it carries in inflammatory reaction. Now the last one, which is the archibacterial ether lipid. I mentioned that the complexity of it tells you that it requires high amount of energy to be broken down, and these ether lipids are found in the cell in the cell membrane of thermophilic bacteria that can stand very high temperatures and very acidic pH conditions, so that even in those conditions, this bacteria still survives and does not get broken down, does not get, the cell does not die in such high temperatures. Sometimes above 100 degrees Celsius, you still will find these bacteria living in those conditions. And you can see the structure is such complex that you have glycerol backbone. Among glycerol backbone, you have some disaccharide, glucose, galactose bound on it, and between these two glycerol backbones, there's one at the end there. You have this branched hydrocarbon, two of them diphatanyl. So these are just carbon, hydrogen, and they are even branched, which they are having some methyl groups on them. So that makes the molecule even much, much bigger and more complex, that it can easily be broken down. On the second glycerol backbone on the right-hand side, you then will have a phosphate group, and then another glycerol that is bound on that phosphate group. That's a glycerol phosphate group which is bound on that. So it's a more complex structure, which is more difficult to break down. That is to say that the melting point of this structure is much, much, much higher, which gives the bacteria such a protection in such high temperatures and at such low pH conditions, acidic conditions. All right, I think this is where I will stop talking, and then I think we still have like 22 minutes, so that if you have anything, you can raise your hand. I will enable your microphone, call your name, you speak, I respond, or the chat is on. You can use the chat, you can write in the chat, and then I will respond to it. The floor is open.